Good morning, ma'am.
Good morning, Namrata.
Okay, so basically the difference is in the sexual reproduction. First thing is that two parents are involved. There are two parents involved in this. One is male and one is female.
Good morning, students. Am I audible?
Yes, ma'am, you're audible. Yes, ma'am.
So, 17, 16 students. That means 6 to 7 students still to join. Was the class going on before this? Yes, there was mushroom organic composting class. Okay, so I think we can start because it's already 21. So yesterday we discussed the diagnosis, no, not the diagnosis, but the management, and we discussed the management of aquatic viruses, how management can be done. So I was going through the management of animal viruses. I thought I'll take one lecture, but the management of animal viruses is also similar. We go for the vaccinations, and the vaccinations are recommended for many animal viral diseases. So it's not I mean same, right? It's same. There's no not any difference like in the diagnosis also. So I thought we should leave that. And now, since most of the syllabus is covered, so in the beginning also I told you that we'll cover some sub-viral agents also that which cause diseases. And yes, a lot of papers are there, but not much things are available, or we don't know much about these pathogens. Yes, of course, about virus we know many things, but then if we talk about prions, we don't know much about prions still. And virophages are also being discovered. A few virophages are being discovered, and but they are not, again, their number is not like in many numbers are not being discovered. Then baculoviruses, you already know, they are being used as a biocontrol agents or a biopesticides. Then there are certain mitoviruses also. So yes, on mitoviruses also there are certain papers which are available, but not much data is being available, right? Then some satellite viruses or the satellite particles. So in plant viruses, sometimes the subviral particles, we call them as a satellite particles, alpha satellite and the beta satellite, they are present, and they don't have any direct role in the disease modulation in the plant or disease in a plant, but their presence definitely enhance the symptom production in the plant. So let's just go through these topics just like that, so that you know about these things also. And then from next Monday onwards, we'll start the revision also, because the course is complete. We have done everything which was mentioned in the course. So we can just in one lecture or a two lecture we can discuss, or we can go for the revision also, and we can see what topics can be revised. So you all can also tell me if there is a clarification required in some topic, or just from the starting of the course we can revise. So this will further help. There was one student who joined very late. I don't know if she's there today, but that she can also, I mean, revise this way, so things will be more clear to her also, right? So today we are going to just go through a very small but very important topic, which is about prions. I'm not sure if you have heard about this term earlier. So yes, if anyone has heard, or if they know about what are prions, please they can just raise your hand. Have you ever heard of a prion diseases, or what are prions, how they cause diseases? Or this is very new to you? Yes, Manasan.
Ma'am, I know that they are kind of viral proteins, and
Uh-huh.
what we learned was that they caused mad cow disease.
Yeah, you're right, actually. Mad cow disease is a very common disease which was caused by the prions, and at that time when this disease was very prominent in the cows, so they people were not left with any option but to then kill the cows at the end, because those they were not these diseases are not treatable basically. These are the like brain diseases, basically encephalopathies. So these diseases, they are not treatable. Yes, there are certain conditions in humans also. Some diseases are caused by these prions in humans also, but still we don't have any management for these diseases, right? So prions, when we talk about prion, we don't define them under some kind of a pathogen or something like that. It is a type of a protein basically, right? So this is prion is a type of a protein which is abnormally folded, which is misfolded. It is a misfolded form of a protein which is found in the brain. And these diseases, they can affect both humans and animals. And usually it was found that in humans, this disease was transmitted to the humans through the infected meat products. So that is the main cause here. And Jacob disease is the very common disease that we know, it is caused by the prion in the human. So, what causes prion disease? This is still not very clear, but we say that the prion protein, right? When prion protein, which is found on the surface of many cells, it becomes abnormal, right? And it becomes misfolded, and then it leads to a sort of a chain reaction when this prion protein, when it comes in contact with the other healthy proteins, it changes those normal protein also into the prion protein. So a chain reaction is being triggered somewhere, and that lead to a misfolded form of the misfolded protein in the brain, and which can cause a memory impairment, and sometimes the difficulty in coordination or the movement. So such are the symptoms, like and especially the people who are affected by the dementia like diseases, or it is also, dementia is not a disease, we can say it is a condition, right? So it is basically a term which is used for old people who have difficulty to remember things, or even to take decision in the normal everyday activities, right? So Alzheimer's disease is one of the most common type of a dementia. So people who have a certain dementia like disorders or conditions, they are more prone to the prion diseases. They are found to be more affected by these kind of a disease, right? So prion is basically a protein infection is the term which we use for this. We don't say it's a pathogen, we say it's a misfolded form of a protein, and which lead to a disease kind of a condition, right? So a prion is an infectious agent which is composed of a protein in a misfolded form. It don't have any nucleic acid, and the word prion was coined by Stanley B. Prusner in 1982. And again, this means a protein infection. So this prion itself means a protein infection. So all known prion diseases, they affect the structure of the brain or other neural tissues and are currently untreatable and universally fatal. So these diseases, we don't have any treatment available, and they are fatal also. So they basically cause neurodegenerative diseases which form amyloids within the central nervous system, which disrupts the normal tissue structures. Now, this disruption of the tissue is basically characterized by the holes in the tissue which leads to a spongy architecture due to the vacuole formation in the neurons. So once these kind of a symptom appears, the disease progresses very rapidly and it leads to brain damage and the death. All known prion diseases are collectively known as a transmissible spongiform encephalopathies, TSEs, and these are not treatable and they are fatal also. So this shows the cross section of a normal brain, and here shows the cross section of a brain with a vacuole formation or a spongy structure due to the prion infection. So if we talk about these diseases in animals, mad cow disease is a very common disease. We all hear. Then scrapey disease in a sheep and a goat. Again, a mad sheep disease. It's called scrapey because these goats and the sheep, they keep on scratching against trees or against the branches, which even scratch their skin also, right? Then we have a transmissible mint encephalopathy, wasting disease of a deer. And in humans, there are a number of diseases such as a Kuru disease and the Jacob disease and fatal familial insomnia. And we have a Justman-Stroessler syndrome also, right? So these are very fatal diseases, and these all are commonly come under transmissible spongiform encephalopathy. So common term is basically TSC for these diseases. So these are some of the pictorial representation where they say the family that cannot sleep, then there's a brain shrinkage and deterioration occurs due to this disease, right? Fatal familial insomnia, people are not able to see, and there's a Jacob disease, which are the human diseases which are caused by the prions. So they say symptoms of a Kuru disease is like a coordination problems, which eventually becomes severe. So what is coordination problem? What do you understand? That you are not able, for example, you want to pick one bottle or a glass, but then your brain and your hand coordination is not very good, and you might not be able to pick that glass, or I mean, whatever you want to do, you're not able to do that properly. So there's a coordination problem. The difficulty in walking, swallowing difficulty, tremors, and the muscle jerks, mood swings, and the laughter. So the brain, the most infected part of the body in this disease. This disease was first noted in the four tribe of New Guinea, Australia, in early 1900s, and they found it that this was transmitted by mortuary cannibalism, and this disease is known as a laughing sickness also because the posture of your face is becomes such that it appears that you're laughing though you are not laughing, so it was called as a laughing disease also. um, Prion, so what happens is prion proteins, they are naturally present in the cell membranes of the brain cells. So normal prion proteins, they are naturally present in the cell membranes of the brain cell, and their role could be in some signal transcription there, right? They play some role. So what happens when a prion enters a healthy organism? It induces the just a moment. It induces the healthy proteins also into the prion proteins, or a diseased form or a misfolded form of a protein, right? So when a prion enters a healthy organism, it induces existing properly folded proteins to convert it into the disease-associated prion form. So the prion act as a template to guide the misfolding of a more protein to prion form. So it is kind of a chain reaction. Say it will keep on converting the healthy protein into the misfolded form. So these newly formed prions can then go on to convert more proteins themselves. So this triggers a chain reaction that produces large amount of the prion forms. This is how they cause diseases, prions. This protein, it polymerizes into the rod shaped structures, and this altered structure is very stable, and it gets accumulated in the infected tissues and can lead to a tissue damage and the cell death also. These prions, they are resistant to denaturation by chemicals and a physical agent, making disposable and containment of these particles very difficult. And misfolded proteins, they do not get attached to the membrane, but they aggregate, they accumulate together, right? So initiation of a misfolding is still not very clear that how it started, right? So it is said that once the infected protein will come in contact with the healthy one, it will convert into the misfolded form. But then how the first infection was started, how the first protein become the prion infected, right? From where the chain reaction started. So this is still not very clear. So all known mammalian prion diseases are caused by the so-called prion protein, which are designated as PRP. And the endogenous properly folded form is denoted as PRPC for a common or a cellular form, while the disease-linked misfolded form is denoted as a PRPSC. SC means scrapey. So after one of the disease which is linked to the prion neurodegeneration. So this is how we represent them. PRPC means cellular or common form, and PRPSC means the diseased one. So this Stanley B. Kusner of University in California, he verified the hypothetical infection prion causing diseases in Scrapey and Jacob disease, and he found that they resisted the ionizing radiation, and the infectious agent, they consist mainly of a specific proteins, though they did not managed to isolate the protein until 2 years after the prisoner's announcement. So it took them almost 2 years to isolate this protein. The prisoner coined the term prion as the name for infectious agent and a prion protein. And he was awarded Nobel Prize in Physiology and Medicine in 1997 for his research into the prions. The protein that prions are made up of is found throughout the body, even in a healthy people. And PRP found in infectious animal has a different structure. And this the infected form or a misfolded form of a protein is resistant to the proteases also. While PRPC is a structurally well-defined, PRPSC is certainly polydispersed and defined at a very poor level. We don't know their structure, very thoroughly still. Then PRP can be induced to fold into other more or less well-formed defined isoforms in vitro. So these PRP proteins can be induced to fold into more or less well-defined similar forms in vitro. It can be done. So this is the heterodimer model which is proposed for the prion application. So here also we can see that there is a if infected one, it comes in contact with the healthy one, it will lead to the conversion of the normal protein into the infected protein. So this is kind of a reaction or a chain reaction which leads to the misfolding of a more and more protein. So and then they suggest that the infected form of a protein exists only in the aggregated form such as amyloid, and that prion replication involves again their replication require at least one infected prion forms. So after then only they will be keep on replicating. Current research suggests that primary method of infection in animal is through ingestion, and it is thought that prions may be deposited in the environment through the remains of the dead animals via urine, saliva, and other body fluids from where it is entering the human body also. So they may then remain in the soil by binding to the clay and other minerals. So if something infected with these animals who have this kind of they remain are in the saliva and other body fluids, or they remain in the soil in some form, it goes to the human body, it leads to the further infection. Recent studies have shown that infection can occur from prions in the manure also. Since manure is present in many areas along water reservoirs or crop fields, it raises the possibility of widespread transmission. So this could be the reason that how it is being transmitted. Then these are the number of the questions which are still not answered. How and why this kind of a misfolding occurs? What triggers this kind of a misfolding? Why it is taking place? We don't know. How then another important point is that how this infected protein crosses the blood-brain barrier. So they say it could be through the endocytosis. That is the possible explanation, but it is still not very clear how PRPSC, since the misfolded form of a protein is same, right, infected one, then how there are different symptoms in the different diseases if it is a similar misfolded protein. So this is still not clear. And is there something else other than the PRPSC that is required for a prion disease to be transmissible? So there are many questions which are still to be answered. There are many things which are still not very clear about the prion. These diseases are though not very common, only, in few numbers they are being found in only few people around the world. So not very common diseases, but yes, if these kind of a diseases they occur, then definitely they are very fatal because we don't have any kind of a management strategies for them, because when we don't understand the pathogen which is causing the disease properly, then it is very difficult to look for the strategies which can manage that particular disease, right? So this is all for today's topic. I just wanted to just a brief small topic on the prions. So just try to read more about the prions, how they are causing. Is there any new something new research has come up for the where it can explain the replication of the prions or any other new idea or a new experiment some people are working. Some there are only few people in the world who are working in this area. Not many people are working in this prion diseases, this kind of area, right? So that's all for prions, a very small topic. And many students they joined very late today, and that's why I just blocked the entrance also, since, but I still let you in. Vivek, you were very late. Then I guess Rupal or someone else, Nehal or
Ma'am,
Rupal, I don't know.
I was
Yes.
trying to join earlier, but for some reason my Microsoft Teams was constantly shutting down. So that's why it was not like
Okay.
when I pressed the join button
Because
instead of
I,
join, it was
yeah,
shutting.
I waited for 10:21, I guess, and then after that only I just locked the room, so that, I mean, you take, or you all take permission who are entering late, right? So
Sorry
please
about
make sure that you attend. No, that's okay, that's fine, since your team was not ready. So students, see, we are almost done with the syllabus. Everything is done. So just try to revise the thing, because then next week onwards we'll start revising the topics also. And since tomorrow, not tomorrow, day after tomorrow, we have a class. So you have a practical exam. Please be ready for the practical. Don't miss the exam. All students, please. It is very difficult to take an exam for a single student, right? So I want all of you to give this exam. This is the midterm practical exam. So please don't miss the exam. So okay, now this is all for today. So if there's anything, any thoughts about prions in your brain, so please you can just discuss here.
Ma'am, I had a question regarding the
Yeah.
midterm actually.
हाँ बोलो मानस मनी.
Ma'am, so is there any particular format that you can
No,
tell?
Manaswani, there is no form where there is like usually it's like one experiment or something. I'll ask you to write it, and then there'll be some marks for the practical notebook and some for the Viva. So this is the usual format that we follow every time.
Okay, okay, fine.
So
Thank
just go through your practicals thoroughly. I might not ask you the direct practical to write. I might ask you through some experiment, or maybe I'll ask you to create your own experiment on the basis of the diagnostic techniques and other techniques that we have learned. Right? So basic, if you know about, you've already studied, so it will not be difficult for you guys, right? So it will be that form.
Okay, thank you, ma'am.
Okay. Okay, students, so please switch on your cameras for attendance. I'll close the class here only, and we'll see you meet day after tomorrow, right? So yes, Adish Mishra, yes, I can see your arm. Uh, yeah, okay, fine. Athoiba,
Yeah, ma'am.
yes. Ayushi, Sam,
Present, ma'am.
yes, I can see Ayushi, Sam. I don't see you. Ishita, Janvi, yes, I can see Janvi.
Present, ma'am.
Kshitij, yeah. Manaswani, yes, I can see Manaswani. Namrita, Kshitij, yes, I can see you. Namrita,
Present, ma'am.
Nandini, uh, your screen is on, but still I can't see you. Nehal, Rupal, Nehal, Sahiba,
Present, ma'am. Present, ma'am.
Sanjana, yes, Sahiba, I can see you. Sanjana,
Present, ma'am.
yes, Sanjana, you're there. Shakti,
Present, ma'am.
Shivani Mishra, yes, Shivani is there. Shakti is there. And Tanvi Sharma, Tanvi is there. Tridip, Vanshika,
Present, ma'am.
yeah, Vanshika is there. Vivek, are you there? Vivek?
Ma'am, I'm present.
Yeah, Yukta,
Present, ma'am.
Yuvraj, yes, Yuvraj, Pradyum, Kumar.
Present, ma'am.
Okay. And I guess your Amazon is again open for if you want to opt for offline classes or something like that. So please check that also, right? Okay, so I'll see you on Thursday with your
Sorry,
exam.
ma'am.
Thank
Am
you
I
so much.
audible?
Yes,
Ma'am, I'm
who's this?
present, ma'am. I'm facing network issues. I'm Nehal.
Nehal, yes, Nehal, आपकी आवाज clear नहीं है बेटा. I can't hear you much. Yes, what is it?
Ma'am, I was saying I was facing network issue. I'm present.
Okay, fine. Okay, you can leave the meeting. Thank you.
Thank you.